For this podcast, we're talking to Dr. Jon C. Mirsalis, who is a Vice President of Translational Development in the Biosciences Division at SRI International in Menlo Park, California. Dr. Mirsalis is an internationally recognized expert in the development of drugs for infectious diseases. He manages two large programs for the National Institute of Allergy and Infectious Diseases for the development of promising therapeutics for the prevention and treatment of a broad range of infectious diseases, including TB, malaria, HIV, influenza, polio, anthrax, plague, Ebola, and COVID-19.
He has personally been involved in the development of over 80 therapeutics that have entered clinical trials, and several have reached the market. He is the author of over 200 publications and abstracts. Dr. Mirsalis received his Bachelor's of Science degree in Zoology and Molecular Biology from Kent State University in Ohio, his Master's of Science degree in Genetics from North Carolina State University, and holds PhD degrees in Toxicology and Genetics from North Carolina State University. Dr. Mirsalis lectures regularly at Stanford University on biosecurity and at UCSF on drug development. He is a past chair of the board of directors of the California Biomedical Research Association and is currently on the board of the National Association of Biomedical Research. Dr. Mirsalis has been certified by the American Board of Toxicology since 1983.
Welcome to the Peace Antz podcast, Jon. And I'll just mention that we know each other initially through doing Spam-A-Lot together at Woodside Musical Theater. And it was a really fun experience, a really funny play. And Jon is extremely talented in acting and music as well. And really looking forward to talking with you, Jon.
Good. Well, the irony is that, we're gonna talk, I think, mostly about science, but and I've been running the infectious disease drug development program for Tony Fauci's National Institute of Allergy and Infectious Disease for many years. Going back into the 1990s. But I am better known for my motion picture activities. I have done projects with 5 Oscar winners, 3 Emmy winners, a couple Golden Globe winners. And when New York Times called me for an interview, a Pulitzer Prize winning reporter called me for an interview, it wasn't about work we were doing on drug development. It was about me finding a lost reel of a Laurel and Hardy film. So I live this bizarre dual life where on weekdays from 8 to 5, I'm a scientist and toxicologist, and then after hours, I'm well known in especially the classic motion picture film preservation industry. So it makes me kind of schizophrenic, and I don't know what I am.
Yeah, no, I was wondering, do you find yourself feeling a little schizophrenic, like you feel like you're almost a different person when you're in different environments?
Well, it's interesting that you say that, because one of the things, this is sort of a broader issue. I'm a scientist by training, obviously, but a lot of the skills that you use in science, data management, data organization, digging for the truth, making things presentable. Those are the same skills you use in film preservation, film archiving. I've been involved in a lot of databasing projects and finding lost films. I've done work with the Library of Congress Motion Picture Division on some of this. And it's all the same skill set. You need good organizational and data management skills, and you need to be able to hunt for clues. And this, scientists are pretty good at that.
So does do you ever find that one side, the musical side, informs the science side and vice versa? In your work?
I don't know if I would use the word in one informs the other. You learn skills. I give a pretty good seminar. I tend to give a pretty good, both an entertaining and a smooth seminar, and I'm a fairly good public speaker. And some of my scientific colleagues who've kind of who struggle with this sometimes, they just say, well, I wasn't trained to do this. How do you give a good talk? And I said, I'll give you the key: musical theater. I said, musical theater, it teaches you timing. It teaches you sort of rhythm. It teaches you the value of how to deliver a punchline. And I said, these are all skills that are useful in presenting. It allows you, it teaches you to speak clearly, to enunciate, to how to not to step on other people's lines. So it's, in other words, I'm only half kidding. I mean, I'm mostly not kidding when I say that, basically for people who have had trouble with public speaking, I say do theater. Theater will improve your ability to interact with an audience, to know how to do delivery, to know how to work within a team and present. I said these are all valuable skills that you get. So, and music is much the same way. I mean, music teaches you timing. It teaches you, rhythm. There's a lot of things you can learn, and of course, science. I guess the flip side of that, science teaches you how to be methodological. It teaches you how to assemble data to create a story, and those are useful skills that we live in, unfortunately, kind of anti-science times. We've come out of 4 years of a very anti-science administration, and it's everybody needs to learn the scientific process, to question when something sounds kind of goofy, crazy. You should have the skills to say, yeah, science, the way I would look the facts on this, science tells me this doesn't make any sense. And, so those, it's all good skills to have. This is why everybody coming up through school should learn some arts, should learn some science, should learn some economics, regardless of what their careers are going to be. That, I'm a strong believer in a well-rounded education, because all these things can be brought to bear to whatever you end up doing in life to help you do it better.
Yeah, definitely.
To go back on what you were saying.
Yeah.
Would you, some of the skills you talked about with theater, would you say that's like emotional intelligence on how to work with people in real time? And because that is something that's hard to cultivate in today's day and age of learning, is emotional intelligence.
To some extent, that's true. I mean, when you're in theater, you have to learn to work in a team. You can be, and again, in music, I mean, I happen to be primarily a solo musician. I mean, I've done, actually, I'm on IMDb. I've created scores for films. I've done silent film scores. I've worked in some documentaries. I do that as a solo, but what theater teaches you is how to interact in a team. It's not all about you. And so in that sense, I think yes, that's correct. It teaches you that, hey, it's not all about you. You're a piece of machinery here. You're one of the cogs in the machine. And if you're not working right, you're going to, if you're blowing your lines, you're going to mess the next person up. If you're not singing harmony, if you're singing too loud, too soft, if you're a musician, that just messes up the whole thing. So yeah, in that sense, I don't know that I would quite call it, emotional intelligence, but it's the ability to work in groups, and which again is a good skill. It's something that women are usually better at than men, and something that men probably have to put a little more effort into learning.
I agree with that. I mean, so I agree with, I kind of like the angle of evolutionary biology, and how women may have been, more social, taking care of the kids and everything, while men went hunting and were more, well, I guess men would work as a team too. So I guess that theory doesn't really make sense. But so
right, I mean, you're getting into some. I mean, this is way outside of the of this topic today. Whatever the topic of it today is, but I mean, sociobiology, talks about that. That they said, not just in people but in other species. I mean, men act the way they do because that was a successful evolutionary development that allowed the species to survive. Now we can argue that some of the ways men act is maybe not the best for society today, and a lot of what I would call men apologists say, yeah, but that's hardwired in, that's in their genes, it's in sociobiology, nothing we can do about it. Well, you can, I mean, because we also have the ability to kind of override those the kind of base instincts to take a club and club something to death that you're having difficulties with. So or drag the woman by the hair and pull her into your cave. I mean, yeah, they probably did that once upon a time. I think we could probably get over that. But
So where are you on the
whole
nature,
different field of study.
where are you on the nature versus nurture like spectrum of belief?
It is well. So I have two PhDs. One of them's in genetics.
Yeah.
So I mean, a lot of it is genetic, but what genetics does is it sets what I would call the background tableau on which all the environmental stuff is built. It's clearly both. I mean, there's mountains of evidence and thousands of studies that show that there are some things with a very strong genetic component. There are some things with a very strong environmental component, and there's a whole bunch of things that sit in the middle that are a combination.
So my understanding is like we have a ton of genes, and a lot of which don't express themselves, but can be activated with different environmental factors. Is that some like what?
Well,
what is it?
yes. I mean, the honest answer to that is a little more complicated, but you've got an estimates, depending whose estimates you believe, but we probably have about 30,000 genes. One of the real surprises of the Human Genome Project is, we knew that mice and rats had about 30,000 genes. We said, well, people are more sophisticated. They're going to have 60, 70, 80, 100,000 genes. And then it turns out we got about the same as mice and rats. And a significant number of those genes are largely the same with the same function. We all have pretty much the same gene to make hemoglobin. But those genes, you have genes turned on and off at different times in life. You have genes turned on and off in different cells. So, as an example, I mean, every cell in your body, and by the way, almost everything I say today is going to be slightly wrong, but mostly right, because everything is a slight oversimplification.
I'm into simplification, so it's fine with me.
pretty much every cell in your body has the same set of chromosomes, has the same genome. And so that means your red blood cells carry the genes for the eye pigment that colors your eyes.
Yes.
And those genes in white blood cells are turned off. But they're in the early days, when we really started to develop more molecular biology and gene sequencing, as you get into the into the late 70s and into the 1980s, they started realizing, they discovered what they called leaky genes, where they would find, and don't quote me on this number, but it was something like 1 in every 10,000 white blood cells, the gene for eye color pigment is turned on. So, 1 out of every 10,000 white blood cells has blue eyes. I mean, it's,
that's
and,
so
and
weird.
this is just, and this kind of stuff happens.
Sometimes,
and of course,
genes get turned on.
Well, it can get turned on due to genetic errors, due to mutations, there are downstream genes that we call regulatory genes. Something can mess up a regulatory gene. And you are just sitting here right now, we're all mutating our genes.
Right.
The mutation rate again, 1 out of 10 to the 6th, 10 to the 7th. So 1 out of every million, 10 million, genes at any time are going to mutate. Now, the more you're exposed to environmental mutagens, radiation, mutagenic compounds, that number is going to go up. The longer you live, that number is going to go up. And because you accumulate these over time.
Is Wi-Fi, would that increase genetic mutation? Like just having Wi-Fi around?
There's no evidence that Wi-Fi does that. I mean, there have been studies on power lines, on cell phones, on a long list of things. The data are all
long-term. You need long-term studies, right? The
data, they've done long-term studies. The data are really sketchy. I mean, there was a big concern in the early days of computers. There was a study that came out of, and I think it actually came out of San Francisco, that said, and they looked at women who used, word processing equipment. In the old days they had typewriters, and then and then they began moving to, the computer monitors. And the early computer monitors, they were these monochrome monitors that didn't have a lot of shielding, and they were putting out a lot of electromagnetic fields and whatnot. There was a study that showed, and it was statistically significant, that women who used these more than X hours a day had a higher risk of miscarriages and babies born with birth defects. And everyone was, oh, horrors, oh my gosh, oh, that's terrible. And then other groups went out to try and duplicate it, and there were like about 20 to 25 more studies done, and no one could duplicate it. And so, we get into statistics, and we always use what we call the p less than 0.05, which is 1 in 20, 1 in 20 chance. And there are a lot of things that happen in a less than 1 in 20 chance. Yeah. And they just happen, and it's bad luck. But if you can't reproduce that, then then it's you just shrug your shoulders and say, well, there's not really a good basis for this working. I mean, to bring this back to COVID, were there cases of people who got hydroxychloroquine who got better? Absolutely. And not to bring politics into this, but people would say, yeah, hydroxychloroquine, it's a miracle drug. But when they did properly conducted, blinded clinical trials, it didn't work.
Right. It was not very effective.
The people who got better, I don't dispute that they got better, but they may have gotten better having nothing to do with hydroxychloroquine, or hydroxychloroquine worked for a very small percentage of people. But again, doing a properly conducted scientific study, and I think there's been several now on hydroxychloroquine, they just said, no, it really didn't provide any benefit. And so in other words, that's the difference between science and anecdote. I mean, the other example I always give,
repeatability.
Something like 1% of all children who are born in the U.S. today will develop some signs that'll put them on with an autism spectrum disorder. Around, numbers vary, but say around 1%. So that's just a number. Ergo, about 1% of children who get vaccinated are going to develop autism. 1% of children who don't get vaccinated are going to develop autism. So if you're a parent with a child who has autism, I feel for them. I mean, it's some of the more severe cases can be pretty debilitating and life-changing, not in a good way. Yeah. You understand that those parents are angry. Their child developed autism, and gosh, they got their child vaccinated. Maybe it's due to vaccines. But you do properly controlled studies comparing, autism rates in vaccinated versus non-vaccinated populations, and there's no difference. That doesn't change the fact there's a whole bunch of people who are anti-vaxxers who are convinced that autism, is caused by vaccines. There's no evidence to support it.
Right.
In fact, the one paper that came out of England many years ago that claimed to have evidence, it turned out a lot of it was falsified. The doctor subsequently, they retracted the paper. The doctor had his medical license taken away because it was all bogus. But once you believe, and again, if you have a child with autism, that changes your life in a bad way. And those people are going to believe to their dying day that this was caused by those vaccinations they got. But that's what science does. Science, asks the questions in non-emotional, non-anecdotal ways. You say, let's take a large group of people and let's look at the rates. And if you can't prove it that way, then it's not real.
It tries to take all the human bias out of, as much as possible out of it.
Right.
I wanted to point out something you talked about earlier about, in our society today, we're not trusting science as much before. And I think we've lost an ability to research. And I think, how would you think we could improve upon this in the schools, or how do you think we can improve upon this as a skill for the common citizen?
So that's a really good question. Really, in fact, I would say that's the $64,000 question of the day. I mean, We need better science education in schools. I mean, simply more education helps. I mean, people who believe conspiracy theories, regardless of what kind of conspiracy theory it is, whether it's a conspiracy theory about the election, or whether it's a conspiracy theory about whether we actually walked on the moon, or whether it was faked in a movie studio. I know there's all these different kinds of conspiracy theories, and there's actually been studies showing who believes them. The basic underlying fact is the less education you have, the more prone you are to believe these. And the more education you have, the more you kind of critically challenge and say, well, if this theory is right, then, and you can develop a hypothesis and you go, oh, wait a minute, but that's not true. I mean, a really simple example is, if you believe that a pizza parlor has a basement where children are being, bled dry by pedophiles. Well, the first question you ought to ask her is, does that restaurant actually have a basement? And when you find out it doesn't, that your theory probably isn't going to hold up really well.
Yeah, you may have heard it from a not so trustworthy source, right?
Yeah, people. So people, I mean, the term, the last 4 years, people use the term fake news a lot, and the term fake news gets used about 95% more than it should. But there is fake news. There is stuff that gets reported that's just not true. And what's more, the people reporting it pretty much know it's not true, but they're reporting it to to make the case that to support their position. And they figure that most people won't bother to dig into the science behind it, and they're right.
Yeah. I think, but so we're talking
that is
about a few things.
The main point there.
The mainstream is, I don't think they want people to pay attention to the science. Like, they just want you to eat up what they have to say, right? And push their agenda, I guess.
Well, I wouldn't say that's true in the mainstream. I mean, the mainstream media gets a lot of bad press, but actually, mainstream media, the major networks, ABC, NBC, CBS, CNN, the major news outlets, New York Times, Washington Post, they have fact checkers. They don't, they sometimes get it wrong, but most of the time when they get it wrong, it turns out that, well, there was conflicting information, and they sort of presented it, it was a little bit of what I would call a gray story. They didn't quite have all the details and maybe announced prematurely, but they mostly get it right. But on the other hand, they have to simplify it for the average reader. There are a lot of things when you get into science where the actual answer is very complicated. And and it's hard to explain complicated things to people. People want a really simple, something that in a 30-second news lead in on the nightly news, or in the first paragraph of a newspaper article, you want to get it. There are things that are, and this is true in science when we talk about diseases and things like that. I mean, diseases are really complicated. Most diseases are complicated and multifactorial, and there's a lot of ifs, ands, and buts. And that's hard for the conventional news media to digest down to level for a basic person. I think most people could understand it. They don't have the patience to sit and listen to all the details.
It's a lot of information, right?
Yeah. But I was thinking we should have some sort of a national Bill Nye the Science Guy. I guess you kind of have that on YouTube with like Smarter Every Day and Veritasium channels and stuff like that. But it'd be cool to have something, and they do actually bring them out for like national events and stuff like that occasionally. So yeah, I think stuff like that is really cool.
Yeah, I mean, I don't, there's not a simple answer to what to do to bring the back. The underlying problem is getting the public back to a place where they can listen to and understand data, regardless of what the data is, whether it's data on COVID, data on elections, data on anything else, to understand that and to have the critical thinking skills to, especially with things like conspiracy theories, most of which are pretty ridiculous. And anyone who had, who would simply take 5 minutes to think about the science would realize, gee, this thing that they're proposing makes no sense and is crazy.
But there are conspiracy theories that are true too. Some, a lot smaller, but it does exist.
It depends. I heard there was an interesting thing. I don't remember if it was 538 or the Weeds, one of these online things. They did a whole show on this. They had an expert on this, and they looked at different conspiracy theories, and they said the most believed conspiracy theory in the U.S. today is that Oswald didn't assassinate Kennedy. They said that was most, and that was believed by something like, almost 50% of the public.
I kind of believe that too.
Just Oswald.
That bullet, something about the bullet going crazy.
The magic bullet that stopped for 1 second, his body went through. Nevertheless, I'll be honest and say I would probably put myself in that category. I don't know who assassinated Kennedy, but the physics tell me that a lone gunman doesn't make a lot of sense. Nevertheless, the data and the evidence, all the reports and all the commissions come to that conclusion, and it's easy to be an armchair general and say, well, I think they're wrong. Well, but I didn't have access to all the classified data and everything, and the average person doesn't. So when you say some conspiracy theories are true, I mean, not a lot.
Yeah, no, totally. But I think the heart of the issue is has to do with trust, public trust in government, in healthcare. There's no trust there, and I think it's because profit and just the way our economy works, profit is kind of number one over people. It tends to be the case.
Well, and I think that's true. I mean, I think there's distrust in a lot of government. There's distrust in various institutions. I think what COVID has I hope has taught people is that the pharmaceutical industry is portrayed as this monolithic, greedy, horrible, parasitic disease on society. If not for the pharmaceutical industry, we would be in a world of hurt.
Yes.
Right now. And I mean, we are in a world of hurt, but it would be far worse because they had the resources and the incentive to drop what they were doing and get vaccines out in extraordinarily fast, I mean, literally record times. And so anyway, let me finish. In other words, so and I'll be honest and say that without naming specific companies, some of whom I have friends work a number of these companies and I've interacted with them in the past, but some of the companies, they probably did it for PR. I mean, some of these companies, they're not going to make money on these vaccines, but they have the sense to say, this is really going to burnish our credentials with the public. Yeah. And to that extent, I think it's true. So, I mean, you do need a functioning pharmaceutical industry to handle things like this. And thanks to the pharmaceutical industry, life expectancy has increased, 25 to 30 years over the last, half to 2/3 of a century, because of, there are cancer, testicular cancer, I'll use an example. Testicular cancer used to be a death sentence. Men got it, and they're usually in their 20s or 30s, and the survival rate was low single digits. Well, I mean, I personally know people who had testicular cancer with metastases to 7 different organs, that was 30 years ago, and they're still doing fine. I mean, Lance Armstrong, forget the doping piece of it. He won the Tour de France multiple times after having, multiple metastases from testicular cancer that before an era when we had drugs for it would have been dead before he ever competed. So, I mean, in other words, modern medicine has done a huge amount, and it's expensive to do it. It's hard to do it. And there has to be some incentive to make money on that. So now, at the same time, I think a lot of pharmaceutical companies love to gouge prices because they can. But it's, I would hope coming out of this, people have a little more nuanced view and say, well, the pharma industry, they push a little too hard on the profits, and we probably need to do something to to make sure there's some reasonable price controls on things, but they're not this horrible, evil, monster devouring society the way a lot of people portray them. They're in business to make money, but they happen to save people's lives while doing it.
Yeah, things are multi-sided, and since it's so big, so you have a lot of people doing fantastic, amazing, just stellar work, and then you have people, taking advantage of other people, because that's, people will do that once in a while. But so I was actually going to say,
even HIV, right? HIV, I think, I don't know why about that. HIV is manageable, right? Now.
Well, HIV, that's an interesting story where, we had, that was almost a record from the time the disease first appeared in the US to the time they had the first approved drug was 7 years, which is at the time, I believe, was the fastest drug approval ever. Ever in the modern era, certainly. But that was only because AZT happened to be sitting on a shelf at the company Burroughs Wellcome, who later became part of GlaxoSmithKline. I think they developed it for cancer, and it failed for cancer. But when HIV came along, they said, well, do we have any drugs that will work for this? In fact, I think the drug was approved within about 2 to 3 years of the identification of the virus. So it's really even faster than it sounds. But that was a case where pharma has this. So again, getting back to COVID, SARS-CoV-2, the virus came along. I don't know exactly what these companies did because a lot of it's proprietary, but what I suspect, the Pfizers and Mercks and J&J and the others did, vaccines is one thing, but there's a lot going on in drug development. A major company, they probably have a drug library of 10 to 15 million drugs, and they have robots, and they developed a method for culturing it, putting it into cells, and they just turned on the robots and they started screening millions of compounds. And they said, oh, well, we've got 2,000 things that kill SARS-CoV-2 in culture. And then, you narrow that down, and then you looked up. In other words, this is the way drug discovery works, is you start by brute force, and then you start putting some, intelligent design into it to say, let's try and optimize this. And then you move from cell cultures models and from animal models to people. And, if you have that infrastructure, you can respond quickly to swine flu, to Zika virus, to Ebola, to SARS-CoV-2, which are all things in the past, 10 years or 10, 12 years have all kind of emerged.
Part of that infrastructure is the research institutes. And so for COVID, I believe it was we have Kizzmekia Corbett, who started researching the spikes on the COVID virus. And
right. So
years ago, so we had a huge head start on developing the vaccine.
Right. So Kizzmekia Corbett, she's actually one of my heroes. She's I don't know her personally, but I know who she is. She works at what's called the VRC, the Vaccine Research Center at National Institute of Allergy and Infectious Disease. And in 2014, she went there. I think she came right out of a PhD and went there and was doing, I'm guessing it was a postdoctoral fellowship. And you typically pick a research project. And she said, I want to research coronaviruses. Well, at the time, SARS was about 10 years behind in the rear view mirror. I don't think MERS had really emerged yet. And people probably looked at her like she was nuts and said, well, why do you care about coronaviruses? But, she was the one that found a way to isolate sort of the spike proteins and make them stable. She showed that you can make a messenger RNA of a spike protein, and that's a good way to cause what they call an immunogenic response. In other words, basically do what a vaccine wants your body to do, make antibodies. And there was this little company, not well-known company called Moderna that had a technology in mRNA vaccines. And she, some years ago, had said, hey, are you interested in collaborating with me in this project? Let's see if we can make vaccines out of mRNAs. And that's how we got from the time the viral sequence was identified to the time the first phase 1 trial started was 66 days. I mean, that would have normally been 3 years.
Right.
But because she had about 6 years of work, behind her on that, that's what allowed us to respond super, super quickly with this vaccine development, which is why the Moderna and Pfizer vaccines sort of made it across the finish line first, and that's the primary ones now. The AstraZeneca and the J&J and some of the others are going to be following pretty soon, we think. But that's how the mRNA vaccines, got there first, because we had in place a good, research infrastructure doing this, and you had Moderna, who was a company who had the resources and the platform. So they had already, taken this mRNA platform into clinical trials. So, that saved a couple of years that we knew that. And that's what led to, sort of this extraordinarily rapid turnaround. I mean, it's just, it's astonishing how quick it was, but which shows, again, underscores, the messages, we need a strong, a very robust basic research program in place, for a lot of diseases, but especially infectious diseases, which can hit very, very quickly. I mean, another story, I was involved when Ebola broke in the US in 2014. And Ebola had not gotten a lot of support. Again, Ebola that's something that poor people in Africa get. And they don't have money to buy drugs, so why should we invest in that? So it was this esoteric piece of research, and we were working with a drug called ZMapp. It's a triple monoclonal antibody cocktail. We had the world supply in our freezers, and we were doing some early work, and we were doing some of the early animal model work on this. And we were like 6 days into a study when the guy from Liberia, who is now in Texas, was diagnosed with Ebola. So Ebola was now in the US. And then you recall what happened next is there was like a nurse who was taking care of him, and then she got on an airplane and flew, I think, to New York, and everybody freaked out. Right. And there was this huge concern. So I remember we had what I call the queen mother of all telecons. We were on a call with NIAID, I think the CDC, the FDA. I can't remember who all was on this phone call. There were a million people connecting. And I said, look, what do you want us to do? Tell us what you want to do. Do you want us to stop this study, collect all the samples we've got, and send them to the CDC? And the FDA jumped in before anyone could say anything. And they said, absolutely not. They said, here's what we want you to do. We want you to continue the study you're doing. If you can, extend it. We want to see more doses. We want to see you go longer in time, because this is probably going to be the only data we get before we put this into people. So we continued with the study. That study became the basis of what they call an IND, an investigational new drug application, that usually takes like 3 years to get there. 6 months after we started the study, the IND was filed. And we're working, the manufacturer sent us, you get all these little tiny vials for, like a vial with like 2 mils in it. And you take them out, you take out what you need, and a little bit left, you throw it out. Take the next bottle, throw it out. And I said, don't throw anything out. Stick it all in the freezer. And save everything, which normally we wouldn't do. And when it was all over, I said, we've got, 80, 100, 120 vials, each with a little bit in the bottom. Do you want it? And they said, absolutely. And that was used to treat 9 Americans who'd been infected with Ebola. 8 of them survived.
Wow.
The one who didn't was already in late stage. So that's but again, we were lucky that we were in a situation where, the NIH, in their infinite wisdom, was funding a little bit of research on Ebola. It wasn't a big priority. They were putting more money into malaria than they were, which is also not an issue for Americans, but it's a big issue globally. They're putting more money into malaria and Chagas disease than they were in Ebola. And so, we had that infrastructure, and, it's not just us. I mean, we do a lot of the drug development, but, there's people at universities and research institutes all over the world, doing gene sequencing of these organisms, doing, early discovery. And you need that infrastructure in place if you want to be successful. I don't know if the next pathogen will be a coronavirus. Again, it could be a filovirus, it could be Hanta, it could be Lassa, it could be a new, H1N9. I mean, who knows? Some influenza variant. We don't know, and so we need to be prepared for any and all of these things. And part of the problem is Congress is really good at throwing a lot of fire extinguisher at specific fires.
Yeah.
So, you know, when AIDS was big, they put a ton of money into HIV and ignored everything else. And, you know, when the anthrax letter attacks hit, they suddenly, you know, within a year, they jumped into billions of dollars into biodefense against anthrax plague tularemia. You know, and then Ebola hits in 2014. They put 1.2 billion into Ebola, and I think it was Francis Collins who went before Congress and said, you know, we don't need 1.2 billion, and we can't really spend 1.2 billion. What we needed was 100 million a year for the last 12 years.
Huh.
But you didn't want to do that. That's stable funding. And you know what they did? 2 years later, they took all the Ebola money away and they put it into Zika virus because Zika broke in 2016 in a swing state in a presidential election year. And then after the election and Zika started to burn out, they pulled it away and they put it into drug-resistant bacteria. In other words, this is not the way to run a research program. You know, Congress needs to, you know, Congress and the administration need to say, look, we're going to, for 10 years, we're going to fund drug-resistant bacteria, you know, filoviruses, coronaviruses, influenza viruses. You know, I mean, you know, build your own, whatever you want. A side issue, don't even want to get into at this because it's a whole different topic, is, you know, whether people believe in global warming or not, the insects don't care, and insects are moving north as we get warmer, and that's why we're getting Chagas disease. That's why we got Zika in Florida, mosquito carried, Chagas disease in Texas. And you know, doctors in North America have, for the most part, have never seen a patient with Chagas disease and don't even know what it looks like. It's a major problem in South and Central America. And
global warming would bring diseases.
And so global warming, it's bringing the vectors north that carry these things. Wow. And so we need to be prepared for that. So the way to do this is to just set a 10-year goal. Okay, we're going to spend 10 years funding this, and not say, oh, well, we're going to take this money away this year because there's this other thing we want to put it on. Research doesn't work that way.
Who's leading that effort? Or like, because it seems like we're so reactionary and it took some heroic moments.
Well, who is leading the effort? In theory, that leadership should come from the White House and the leaders of Congress. You know, again, I don't even want to go into how the White House led the coronavirus response, but you know, you need long-term leadership. It's not even, you can't even just blame one politician or one political party. You know, these are 10 to 20-year solutions that we need. You're not, you know, and I would say that not just infectious disease, I would say the same for Alzheimer's. It was the same for going to the moon. You know, the program started under Kennedy and then continued under Johnson, and they landed on the moon under Nixon. I mean, in other words, I mean, you need a national effort where the leaders just say, this is important, I'm going to fund this, and not say, well, that was a pet project of this other party or that other politician, I'm not going to support that. Or worse, I'm going to issue an executive order to undo it. In other words, we ought to collectively sit down and say, here's what we should do. But it's hard to do because, you know, elections are basically every 2 years for Congress, and people want to respond to voters with what the hot button issues are. And, you know, COVID will go away. I mean, it's going to take a long time, but eventually this will disappear and people will begin to forget about it. And I hope that people don't say, well, I guess coronaviruses aren't a problem anymore. Because coronaviruses have been a problem for a number of years, and they're going to continue to be a problem for decades to come. And so we need to just, certain amount of money every year needs to be put into research on coronaviruses.
So for the coronavirus, for the vaccine, the Pfizer and Moderna vaccine have mRNA in lipid balls or fat balls, basically. And I was wondering if you can explain how they make that, or how do you make how do you put our mRNA into a fat ball?
I'm not the best person to answer that. That's more of something to someone who does manufacturing. I mean, at a high level, you make the message. There are ways to make what they call liposomes, which are small, lipid particles.
Oh.
And there's used a lot in drugs now, in drug development. It turns out that it's a really good carrier, and there are projects now where people look at liposomes as as drug delivery devices, and you can get good timed release. I mean, getting back to HIV, in the early days of HIV, and to regress back to a musical theater reference, Sheraj, in Rent, you'll recall that one of the little side plot elements is that in the early days of HIV, people wore timers on their belt, and the in other words, one goes, oh, it's time for my AZT. Oh, it's time for my DDI. Oh, and because some were every 4 hours, some were every 6, some were every 8. Oh. And of course, in Rent, that's the plot element that Mimi's boyfriend finds out she's AZT positive because her timer goes off, and she says, "Oh, AZT break," and that's how he finds out that she, like him, is also HIV positive. But anyway, but that's always a real problem for compliance and medication. So then Gilead got very rich, became, I believe, the most successful or the highest market cap biotech company in the world by simply packaging these drugs in a once a day pill. Put the drugs together, pop one pill a day. Well, there are some people now working with liposomal formulations where it's not a once a day pill, it's a once a month shot.
Right.
Where you can give a subcutaneous or intramuscular injection and have this depot of, as you call them, fat balls that have this drug that just slowly releases into the system. And so you can get, single administrations very far apart. And that improves compliance. That's particularly good when you look at, say, communities in Africa where HIV rates are 100 times higher than they are in the US, where it's just difficult to do distribution. It's difficult to get the medicine in. But if you could have traveling healthcare groups that would go into a village and give everyone who's HIV positive a once-a-month shot and say, well, we'll be back in a month. I mean, that could be a game changer in HIV. So getting back to your question, I mean, on liposomes, the again, the messenger RNA, there are manufacturing processes where you simply get this incorporated into these small little lipid vesicles, and then you give an injection. And then, of course, what happens is the messenger RNA is delivered. It gets into cells. The cells are efficient but stupid. They say, "Oh, here's a messenger RNA. I think I'll translate that into a protein." It translates it into a spike protein, and then that's a foreign protein. And your body says, "There's this foreign protein," and makes antibodies. So that's how it works. Now, you could just give the spike protein. And a couple of the vaccines, that's, I think, the Novavax and the Medicago GlaxoSmithKline, those are protein based. That you just give the spike protein. And then Johnson & Johnson, AstraZeneca, Oxford are working on what's called an adenovirus model, where again you have similar genes. You have the genes for the spike protein, but you can put them into an adenovirus, and the adenovirus basically infects you. It's a very, very mild infection, doesn't really make you sick. But then again, same thing, makes the spike proteins, antibodies are developed against the spike protein, and and it's and then and then you develop immunity through that mechanism.
Gotcha. I remember Dr. Corbett was saying that in South Africa, there's a new strain and the spikes are a little more worrisome. I was wondering what that means.
So yeah, so there's new strains out of the UK, out of South Africa, and out of Brazil, and they're a little worrisome. And I think the South Africa strain in particular, what it means is it's a slight. The nice thing is a lot of these coronaviruses, they have the same spike protein. It's almost exactly the same, and antibodies recognize it. The one out of South Africa, there's been a mutation that's led to a slight change in the spike protein structure. So the antibody, instead of and again we get into the specifics of binding affinity and how
I was
tightly.
actually going to ask you about, how does because Dr. Corbett was mentioning that there's a spring sort of mechanism, and then, for the binding site, and the antibody kind of locks the spring so it doesn't bind.
I mean, I can't comment on. I mean, I don't know all the the protein structure of the spike proteins.
Okay.
She's a real authority on this.
Gotcha.
So whatever she says is going to be more correct than what I say. But an antibody is like a lock and key, and it binds very tightly. And if it binds to that binding protein, then it can't infect the cells. And so, in fact, the antibodies will bind it and basically kill the virus. So what happens is if you change the spike protein a little bit, it might still bind, but instead of completely, totally binding and shutting it down, maybe it binds loosely, or it's a weaker binding, or binds, instead of locking up 99.999% of the binding sites, maybe it only locks up 85%. And so, we think the vaccine will confer some protection against these spike protein variants, but it may not be as efficacious.
Gotcha.
We don't, and right now it's, I think it's a little too early to know for sure. I gave a talk on COVID early in the outbreak last year. I gave one in early February where I said, oh, this is an interesting virus variant we ought to keep our eyes on. And then I gave one in April, and I always had a disclaimer at the beginning of the talk saying, warning, much of what I'm going to tell you is going to turn out to be wrong.
Right.
And that turns out to be true with COVID. I mean, most of the stuff we thought we knew last March turned out to be wrong, and certainly on treatment. And then we get better. But let's face it, this disease is just over a year old. We're still learning things about HIV 40 years later. We got a year on SARS-CoV-2, and we keep learning, and our knowledge keeps evolving, hopefully faster than the virus keeps evolving. But it's this combination. That's why I said we need the good infrastructure to do it. To do the basic research, and then to do the development of products to advance them.
Awesome. So I had kind of a random question. Do you make LD40s for the MSDSs? Is that like part of what you do?
Well, it's not LD40, it's LD50. I mean, it's just,
LD50.
So a little terminology. So MSDS is Material Safety Data Sheet.
Yeah.
All products that enter into commerce in the United States that cross state lines are required to have what they call an MSDS. And that's basically, here's what this material is, here's where it's unsafe, or here's what we know about the safety or about the risks associated with it. For drugs, we tend to do different things for drugs. Drugs have to have safety done, but it veers back to to the discovery phase, and where early on in discovery the chemists are trying to make stuff that works. So they're doing efficacy. They just you start with cells in culture, you move to animal models. So if you take SARS-CoV-2, the first question is, will this thing kill the virus, or if not kill, will it bind up the spike proteins in culture? Yes or no? If it's no, move on. If it's yes, and then you got a bunch of things say, well, gee, these are the 10 best we've got. Will they do that in an animal model? So you get a mouse, and there are transgenic mice that have the ACE2 receptor so that it can infect them. And you can do a study in mice and say, yes, it is protective, or no, it's not. And if it's no, back to the drawing board. Well, then if you show it works, then it sort of moves to development people. And they start asking the same questions. Is this safe to cells in culture? Is this cytotoxic? Does it have binding? There are things called tissue cross-reactivity assays where on things that are binders, antibodies, like monoclonal antibodies, for example, I think like the Regeneron cocktail, and I think Eli Lilly has a monoclonal cocktail. You do panels, people have like panels of frozen human tissue, and they want to know, like cadaver tissue, and they want to see, does this bind to tissues? And they say, ooh, this thing binds very tightly to the liver. That's going to be a problem. So in other words, you keep going steps of the way, and you kill. Our motto is kill early, kill often, because you want to do this before you get into people. Well, then it moves into animal studies. I know a lot of people are very anti-animal research, and if there was an alternative to animal research, everyone would do it, because animal research is hard and it's expensive and it's controversial. And I know a lot of the animal rights groups say, well, you could just do this with computer models. No, you can't, actually. And if we could, we would do it, and we would save millions and millions of dollars. But you can't. So you go to animal models and you determine that if it's safe. Well, then once you've determined that it's safe and you know how to make the stuff, and you do the chemistry to see, and we know there's no toxic impurities in this, and let's say there's going to be pediatric, and you say, well, we're going to give it to kids, but they can't swallow pills, so we have to make it as a cherry syrup. You go through all the steps. Then you go to the FDA and file what's called an investigational new drug application. And then that allows you to test it in people. And now you go into people and you say, does this, is it safe in people? And once you do that, it's a phase 1 trial. Then you do a phase 2 trial and you say, does it work in people? And then you do a phase 2 trial. And then a phase 3 trial is sort of a combination. Is it safe and does it work in people when you do it in large populations at multiple sites?
And that's the hardest one.
And that's the hardest. Well, I mean, I wouldn't say it's the hardest one. Statistically, 1 in 11 drugs that make it to phase 1 make it to market. Drugs that make it to phase 3, 42% make it to market.
Got it. Yeah.
So your odds are still better, but a lot of things fail in phase 3 because and even when it gets more complicated. I was on the FDA over-the-counter review committee when I think it's Prilosec or Prevacid, whichever the one that omeprazole is. It's either I think it's Prilosec. When that went over the counter, I was invited by the FDA to be on the review committee because that was a drug I'd done quite a bit of work on. They asked questions like, is the label clear enough for someone with an 8th grade education to understand? I mean, they in other words, they raised the bar on these things. But anyway, but you go through all those steps, and there's little detours. Like before you can give it to pregnant women, they say, well, let's do this in pregnant rats and see if it has an adverse effect. And the very infamous story, thalidomide was a drug that caused major birth defects in a number of countries. It was given to women. I think it was for either, I forget if it was for people who had trouble sleeping when they were pregnant or for morning sickness, but they gave it to pregnant women. Well, a woman named Frances Kelsey, who was a reviewer at the FDA, she was vilified by the pharmaceutical industry because she said, I am not convinced that we have enough data to say this is safe in pregnant women. So she held up on the approval. And it turned out that when they did the animal studies, they did it in rabbits, thalidomide turned out to have all these horrendous birth defects. It was never approved in the US. She eventually, I think it was President Kennedy, gave her which is the highest civilian, is it the Presidential Medal of Freedom or whichever it is, the highest civilian order you can get. He gave it to her because she kept thalidomide off the US market and prevented, thousands and thousands and thousands of birth defects because she wanted to wait for the animal studies that showed, that confirmed, in this case, confirmed that it was going to have adverse effects. So again, part of what we do is some of that animal testing. And again, I said, I know that's controversial, but if there was a way to do it without, we would do it.
I feel like mice is pretty, that's pretty common. Like people are like, yeah, mice, that's fine.
Well, it's interesting. You always see sort of the fundraising of a picture like a monkey in a cage. Mice and rats account for about 95% of the laboratory animals.
Right. Yeah.
And the same mice and rats, you'd be setting traps to kill them if they were in your home.
Yeah.
And animals, they're treated as humanely as possible in the lab. There's oversight committees. You have to jump through all the hoops to get them approved. But until I hope to see in my lifetime a day when computer models and in vitro testing or organ-on-a-chip systems can replace animal testing. But they were saying that was going to happen when I was a college student a long time ago, and it still hasn't happened because it's hard. It's very, very hard to mimic that. Whereas, rats and mice have essentially all the same organ systems as people. Genetically, they're actually in the areas that count, they're pretty similar. And we stop a lot of drugs. We stop a lot of drugs from getting into people because it's something that causes liver injury in a mouse.
Totally.
Save
plenty of human lives.
That's what you need to that's what you need to do. So.
All right, John. Well, I could ask you questions all day, but yeah, it's 2:30. So,
is there any one thing you wanted to ask me that you didn't get to?
Oh, so many things, John.
Pick one. What's your most burning question we didn't talk about?
The most burning one?
I have one.
Yeah, you do it, Sachin. Take it away.
Something, I'm not sure if this is even in your area of expertise, doctor, but where we live, drugs, I'm talking about street drugs are such an epidemic. When we're talking about epidemics, meth especially, and what is being done, or I feel like, especially in where I live in San Jose, there is nothing being done to tackle this issue. And I used to work with homeless youth, and the addiction rates were so high, and it's just so prevalent. And is there anything that's being done on that front?
Well, on the medical, I mean, I can't speak to much of the problem is a problem of society. I mean, a lot of it is societal problems. Despair, unemployment. These are all things that can lead to it. I'm neither qualified nor willing to bring that up. On the drug side, there is a fair amount. We actually have a contract from the National Institute of Drug Abuse to help with development of sort of countermeasures to help people, to kick them off of heroin, for example. Other thing that's being done there's because of all the very significant number of opiate overdoses. We actually have a program right now that actually Department of Defense is funding that is to look at new drugs and like auto injectors that can be used for better emergency response to treat overdoses. I mean that doesn't address the societal issue. I'm not qualified to do that, but from a medical standpoint, I mean we're getting better and better at overdose treatments. We're getting better at treatment programs and drugs to help people get off. But this is a hard problem to solve, and it's a problem that goes back well over a century. It's hard to even wrap your head around how can we prevent people even just getting started. And then you get into things like, economic opportunities. I think an interesting aside, I don't know if you've seen the data, West Virginia, who I don't think of as a particularly, medically hip state, they have the highest vaccination rate in the nation. And everyone's going, West Virginia? Really?
Right.
Seriously? Not California, not Massachusetts. California is actually terrible, one of the lowest. And my theory is because West Virginia has a very high level of opiate overdose. So they are already primed for kind of emergency medicine, emergency response, things like that. They've got a lot of public health facilities set up to do this. I think it was easy for them to transition that to vaccination. That is just my theory. I don't have any real evidence to support that, but just based on all the pieces of the puzzle, that's one explanation for why West Virginia appears to be doing a very good job, and and they deserve credit for that. And by the way, West Virginia is doing a very good job on addressing opiate addiction. They have a big problem, so I think their numbers aren't good, but the way they're responding to it, I think is pretty effective, and they should be given credit for that. So that maybe that's what helped with COVID. So again, it's one of these you wouldn't have predicted that necessarily, but again, good evidence for why you need broad public health programs, you need good public health infrastructure, you need good research infrastructure. And if you do all this, then we'll be better prepared for the next pandemic or crisis that comes along.
I thought of a question, if I could put one more, John. What do you think of UBI, Universal Basic Income?
Boy, that's really outside the medical. personally, I'm not a fan. I'm not a fan of anything that ends with the words for all, blank for all, Medicare for all, universal income for all, free college for all, because I think it needs to be more targeted. I mean, I would rather, in other words, I mean, under universal basic income, someone would give me, make up a number, $1,000, $2,000 a month.
Yeah, yeah.
I don't need them to give me $2,000 a month. So
Yeah.
don't give me $2,000 a month. Don't give anybody like me $2,000 a month.
Andrew Yang.
They don't need it. Give it, instead, target it to people who are below, and it's like the stimulus checks is another thing. Don't give stimulus checks to people who are working full-time, gainfully employed, making a good income and are above a certain salary range. They don't need it. Give it to the people who are unemployed. So just in general, I mean, I believe I'm for raising the minimum wage. I'm for, basic increased infrastructure for healthcare, for various support things. But don't make it anything that is blank for all, because the all doesn't need it. You just like, I mean, I was, I've been gainfully employed long enough. I was able to send all 3 of my kids to good schools and pay for college and have them graduate with no debt.
So
I don't need college assistance for them. And I shouldn't be taking that money from someone who does, because obviously it's not infinite dollars. And if you give it to everybody, then that's somebody who could have gotten more, who maybe needed more. So that's my long-winded answer to UBI.
How do you feel about the term freedom dividend, to rephrase it, so it doesn't have the for all? So in a sense, it's thinking of it in a different way. You think of it as, for being a citizen of this country, you are considered an investor and you get a dividend for participating.
Again, if it was, it has to be means tested. In other words, if you need it, I'm all for, I'm all for bringing people up to some basic level that assures that they're not living in poverty. In other words, that they have housing, that they have, that they have food, that they can get basic education for the kids. I believe anyone who wants to attend college should be able to attend college. And if they can't afford it, we should find a way to pay for it. I believe everyone should have health care. And if they can't afford it, we need to find a way to pay for it. It's this idea of, well, we're going to give everybody X that I kind of object to, because you're giving to an awful lot of people who don't. I mean, healthcare is the same thing. I'm not opposed to Medicare for all in principle. It's just that you already, I think you have 150 million people who already get employer-sponsored work, healthcare, and 70% of them said they really like it. So politically, any attempt to take that away and replace it with a government-run system is a non-starter, because you're going to have 105 million people who are going to object. And that's, but as I said, politically, there's no point in debating the merits of Medicare for all, because we simply have to say politically it's not ever going to happen for that reason.
Sure.
Now, if you created a government system that was a competitive system that was really good, and people said, oh, I could move that. I mean, I qualify for Medicare now. I'm of an age, I can get Medicare now. I stay with my employer health care because it's better than Medicare. It's a better deal. But give me a really good public option. And then if I'm somebody working at an employer that maybe doesn't have that good, I said, well, I don't need to kick into your crummy plan. I can go get some Medicare for all. Then you've got a competitive system. Now, you may over time move to a Medicare for all if you do it that way, because the majority of people will want it. And if you get to the point where now 50, 60, 70% of the country moves to that, then the private insurance companies probably will affect, I don't know if collapse is the right word, but they will begin to go away, or they will have to lower their prices or improve their benefits dramatically. But again, let the market decide that. But again, don't give stuff to people. Don't spend a lot of energy trying to give stuff to people who don't need it and don't want it.
Well, so UBI, the way Andrew Yang presented it, when he was running for president, it would be an opt-in thing. So it's not forced on you, it's opt-in. So do you want $1,000 a month? Yes. Okay, there you go. If you don't want it, then that's fine.
Well, but because people are naturally greedy, they're all going to opt in.
Right. So I guess it kind of depends a lot on what you think of people. Do you think people are greedy in
Yes,
nature, or do you
I think the vast majority of people are greedy.
But I think the vast majority of people are actually trying to, need meaning in their life and trying to do something, and are actually struggling week to week, paycheck to
Well,
paycheck.
but again, I mean, there's ways to test for that. You take you look at their last
But that
year's.
costs money to test for that.
No, it doesn't. A
No?
computer could do this pretty fast. I mean, here's a simple model for it. With a push of a button, the computer could run every taxpayer in the country and get their 1040 from last year. And I'm making a number. I don't know what the right number or formula is, but let's say if your 1040, if your total adjusted income last year was less than 3 times or 4 times the poverty level for your community, because that number is different in California than it is in Alabama. If it's less than 4 times that, you are automatically sent a check. I would probably be more, I'd like to see the details, but that's something I would consider. But the idea that I mean, I like to pick on me because I'm fun to pick on. The idea that I would say, well, everyone else is asking for their $1,000 a month, so I'm going to ask for it too.
Yeah.
I shouldn't be able to get it, and there shouldn't be a mechanism for the government to get it to me, either automatically or on request. I don't need it. I don't want it. And by giving it to me, and again, I don't say me, but
if you don't need it, then what if they don't sign up for it?
the tens of millions of people who don't need it. Why would we want to spend the money? Because ultimately, nothing is free. Again, when they talk about, free childcare, free healthcare, none of it's free. Somebody's paying for it.
Yeah.
It's just a question of who is paying for it.
Right.
And so again, as long as they're means testing things, well, let's give it to the people who need it, and not to the people who don't need it. And I would say an awful lot of those 150 million people who get employer-provided healthcare, some of them are on the really low end, but most of them don't need it.
I would argue when you means test things and you set a requirement for people to get something, you get artificial, you get people who are not going beyond where they're at because they're getting benefits or something like that, right? I'm not sure if I'm phrasing that properly.
Well, I mean, there will be artifacts. There will absolutely be artifacts, but there's artifacts the other way too. In other words, in one way, the way I'm proposing, some people might fall through the cracks. Absolutely, I acknowledge that, and you need to work very hard to make sure that's not the case. And an appeal system where someone says, "Hey, you've dropped me. You say I'm not qualified, but here's my actual situation." You have an appeals mechanism. But the other way automatically gives to a whole bunch of people who don't need it, which I think is a bigger artifact.
Gotcha.
So, I mean, there's the word need is so people don't need UBI, but I think it will create a cultural shift into a more abundant mentality for mass amounts of people. So I think that's kind of what I'm banking on for
I don't know that there's
effect.
I don't know if there's good evidence to support that, honestly.
Sure.
I mean,
doing studies right now.
I would like to see something put forward. You'd probably, again, because of political reality, you'd have to start modest, and you would start modest with something like, we're going to give it to everybody who's at the poverty level, and you roll that out and show that it works. I mean, even go back to the ACA, Obamacare, in the early days. When they rolled it out at first, it was a mess. And people hated it. The websites didn't work. They didn't know how to administer. Rates were bouncing around from year to year. And then after about 5-6 years, they kind of got it right. And that the program went from being wildly unpopular to so popular that when Republicans had complete control of Congress and all branches of government, they couldn't get rid of it because it was so popular. So you step it up. That's why I said, if you want to head to a Medicare for all, start with a good, well-focused public option and do it really well. And I guarantee the first year people will hate it. Second year people will dislike it. But the third year they'll get some of the bugs out and people say, "Oh, this isn't so bad." And then 5 years later, maybe you can expand it a little.
Is that kind of what California does with Medi-Cal?
Yeah. Well, and Medi-Cal's something different. I mean, California, did set up exchanges under the ACA, like many states did, and have done, have done a reasonably good. I don't participate in that. Again, I don't need it, so I don't have firsthand experience. But in generally, the polls say that it's pretty favorably viewed. I mean, same thing, in Massachusetts, which they jokingly call Romney Care, because Mitt Romney was the one who did it when he was governor, and then after he did the system and said it was great, once he was not in the governor and running for president, he said, "Oh, that system was terrible." But it actually was a pretty good system. But it took him a while to get the bugs worked out. And, any major new overhaul is going to take time.
Start small, huh?
Cool. Well, thanks so much, Jon, for
hey, it was good talking to you guys. And if the timing is right and the subjects are right, maybe I'll come back in the future and we can chat some more.
Awesome.
Oh, I'd love that. That'd be awesome.
Yeah. You have a good weekend.
Take care. Bye-bye.
Thanks, Jon. See you.